And now we have our keynote presentation with Professor at UC Davis, David Block, who has organized several programs there dedicated to improving food systems, human nutrition, and sustainability in agriculture. Professor, thank you for being here, and please take it from here.
Great. Thank you for having me. And thank you, Anna and Kara, for the introduction. Let me see if I can share my screen. All right, thank you. Great, sorry about that, but it's great to be here today to talk to you about our work on cultivated meat at UC Davis. And what I'd like to do today is focus what we're doing on what we are doing at UC Davis in the area of cultivated meat, and then give you kind of a flavor for the specific things that we're working on in my lab and some of the other collaborating labs at UC Davis. So I think a lot of you here, maybe all of you here, have a pretty good idea of what cultivated meat is. And of course, in conventional animal agriculture, we're going from the cow to the meat, or the chicken to the chicken breast, or whatever it is. But of course, in this case, what we're doing is we're going through cells to a fermenter and then to the meat. So, sorry, just trying to figure out how this is working. And so hopefully, if you can't see what's going on in my PowerPoint, you'll let me know, but it looks like it's going okay now. So this is a picture of basically what a process might look like. And of course, a lot of companies are working on the process now. And so this is just an idea of what we think a process might look like at a commercial scale. But going from a vial of frozen cells, whatever those are, in this case, let's say cow cells, through a bunch of stages of growing and growing and growing, getting to larger and larger scales until we get to a production scale, let's say 25,000 liters or more. And at that point, we might differentiate into muscle cells, fat cells, and connective tissue cells. And if it's an unstructured product, we would mix those cells to create that product. And if it's a structured product, that's an extra kind of level of complication where we might do something like 3D printing into a structure to get something like the piece of meat you see here. Now, as we're starting, and as obviously companies are starting in this area, and there's even a product out on the market, the question is, where do we look for inspiration on what these processes are going to look like? And I think a lot of the initial inspiration comes from biopharma, because that's where large-scale cell culture is being done now commercially, and has been for the last 30 or so years. Now, unfortunately, the products coming out of biopharma are being sold on the order of $1 million a pound. Obviously, that's not going to be a viable alternative for the meat industry. And so we have to think to ourselves, how do we get this process to be less expensive? Now, one of the key things that we can do is, instead of looking to the biopharma industry for inspiration, because that's where we're growing animal cells at a large scale, we can actually turn in the other direction to another part of the fermentation industry, and that's where we're producing fermented foods and bulk chemicals through fermentation. This is something I worked on in my career early on in industry. And there, we're creating products through fermentation that are on the order of $10 a pound, or maybe even less. And now this starts to get into the realm of what we want to sell our cultivated meat at, to be competitive with other sources of protein. So these kind of fermentation products are everything from wine and beer to soy sauce, citric acid, xanthan gum, MSG, and so forth, vitamin C. All these products are made wholly or at least partially through fermentation. And so it's possible to do fermentation processing at scale and produce an inexpensive product. That's where we want to look to for our inspiration. So the question is, can we grow animal cells and grow them like we're growing, producing these other products. Why is this important? Well, I think if you didn't know why this was important, you wouldn't be at this meeting. But let me just show this data from Kent Bradford, also from UC Davis. By 2050, we're going to have an increased need of 60% in food over 2005 levels. And obviously, or maybe it's not obvious, but it's hard to imagine expanding conventional animal agriculture that much to meet that kind of need. And so what we need to look at is developing protein alternatives. And we're going to hear a lot about that, or you've heard a lot about that at this conference already, and developing new means for producing meat. And that's what I want to focus on for now, this idea of cultivated meat. Cultivated meat is a hugely growing industry, at least in the number of active companies and the amount of money they're raising. Certainly, hopefully it's here to stay. And I think the next step is, how do we get over the hurdles that are holding back the industry? So the way we've looked at this here, there are really three hurdles to commercializing cultivated meat. One is to come up with cell lines that grow when we need them to grow and differentiate when we need them to differentiate. The second is to come up with inexpensive media to grow the cells and to be able to scale that process up to commercial scale that's a large enough scale to make this financially feasible, and then create a structured product, whether it's steak or a chicken breast or whatever it is. And of course, we have to do all of this and still have it be economically feasible and sustainable. So let me talk about, given these challenges, let me talk about the origins of what we're doing at UC Davis. And this is a timeline. It's a short timeline. It's actually interesting when we think about this, how short a timeline it is. If you look back at other industries like the biopharmaceutical industry, which has similar roots, that industry has been going on through production and research and so forth for almost 40 years now. And this timeline that I have, and of course we weren't the first people working on this, but this timeline is less than 3 years old. Okay, and so we have a lot of work to do to catch up the academic efforts to support this growing industry. So we started at UC Davis working in this area in December of 2018. It was in association with a Food Innovation and Engineering conference that AICHE offered, American Institute of Chemical Engineers. I was one of the chairs of that conference. And a speaker from Good Food Institute got up and spoke about the good work that they're doing. And it was really an inspiration for us, for me and others at UC Davis, to start working in this area. Through the next year, we started getting graduate students, Zach Cosenza and Ted O'Neill in my lab, who both became New Harvest Fellows eventually. And in October of 2019, as Kara said, we started the UC Davis Cultivated Meat Consortium with about 30 researchers on UC Davis's campus who are really very interested in getting into research in this area. We also get GFI funding just after that, which is great, to work on chicken cells in our lab, and Keith Barr's lab, our collaborator, along with Karen McDonald and Soman Nandi at UC Davis. And then in February of 2020, we decided to put together a large-scale proposal through NSF's Growing Convergence to really have a larger effort, really building a critical mass on campus to work through the important hurdles that we talked about. So the NSF program was funded as of October 1st. It was, as we'll see, very early funding, federal funding for this area. And now by June of 2021, about 8 or 9 months after we started the project, we have 13 faculty members on campus, 10 graduate students, about 5 undergrads working on this project, with a lot more coming up with ideas now that we have this critical mass, and starting to apply for all kinds of other funding as well. So just quickly, the NSF program that we applied to is for research driven by specific and compelling problems with deep integration across disciplines. And of course, this is exactly what we need to solve the problems, the hurdles, to get over the hurdles, technical hurdles for cultivated meat. So this project, again, had 13 faculty members. It's for $3.5 million over 5 years with a checkpoint after 2 years. And it is the first federal funding for from the US for cultivated meat. So we're very proud of that. The NSF proposal has 4 objectives, and I have them written out here, but it really comes down to these hurdles. Developing cell lines that are going to grow when we need them to grow and divide when we need them to divide. Inexpensive media, the creating structure. And I will say, in addition to these things, understanding how all these things affect the nutrition, the taste, the stability and shelf life of the product. And then finally, the 4th objective is to continuously be looking at the economic viability with this public domain information and the sustainability, and how does it compare to conventional animal agriculture and other means of producing dietary protein. So it really is a convergence of disciplines. These are the main PIs on our project, and kind of color-coded for their areas. You see there's a lot of different areas here, from food science to engineering and biomedical and biochemical engineering, tissue engineering, muscle physiology, stem cell, economics, and so forth. So it's a very much a convergence of these disciplines to work on this. And I think that's exactly what the industry needs. You can see if we rearrange these into the objectives, and you can see there's more circles here than I showed because some people are working on more than one objective. We're really trying to attack each of these things in a multidisciplinary manner. I think that's going to be the only way to do this efficiently. And so this has worked out really well so far. Having these people from different disciplines work together has created a lot of different interesting approaches and new ideas for research projects. So far it's working really well. Now, in addition to the research that we're doing, there are other things that we're doing as part of this project. First of all, we're training students, graduate students, postdoctoral fellows eventually, other researchers, professional researchers, undergraduates, and these are going to be the really critical employees of companies in the future. They're going to push those companies through the technical hurdles or over the technical hurdles to get products to market. We're also doing monthly seminars on science and technology of cultivated meat production. We've been doing that since last December, and we've had a lot of attendees from UC Davis and beyond, and speakers from UC Davis and beyond, which has been great. We're starting to have opportunities for industry and academia to mix, and we have an advisory board that's made up of people from industry and government, which has been great. And we will be running extension programs to extend our knowledge to industry and training courses for the industry, which is absolutely critical to advance any new field. And as Kara mentioned, we will be offering our first short course on cultivated meat at UC Davis, September 13th through 17th. I have a link there. There's more information online, so go online and look if you're interested. Now, I want to point out one of the important things of working with any university, whether it's UC Davis or any other university, is that especially land-grant universities like UC Davis, ones that are focused on agriculture and food, are really good at helping industries get going and to help them solve their problems. We do this on a regular basis. UC Davis is known for its Viticulture Enology, the main department that I'm in, and helping the industry to grow in California to become world-class in a very short period of time. One of the reasons that happened is because, well, some of the reasons that happened is because the research we did that helped the industry overcome problems. It's because of the students that we trained that are now the backbone of that industry. And it's because of the extension courses that we offer to keep people in industry continually changed. It's something that we're really geared towards doing, and we're hoping to do the same thing for the cultivated meat industry and alternative protein industry. Okay, so that's kind of a little bit about what's going on at UC Davis. In the last 5 minutes or so I want to just kind of give you an example of one of the things that we're doing and directions we're heading. And so I mentioned all the people working on this. I want to just point out, give you an example of a collaboration with Keith Barr, who's a muscle physiologist. He works on muscles and tissue culture. I'd never worked with Keith before this, and so this is kind of a new collaboration, but it's been going really well for a couple of years now. And I want to talk about how do you develop an inexpensive plant-based growth or even differentiation media for cultivated meat cell lines. Well, I think any of you who have tried doing this know that optimizing growth media is difficult. This is an example of DMEM culture media with fetal bovine serum. It's complex. And to actually optimize this for production and make it cheaper is definitely not a simple thing. There's a lot of things to look at. In addition to the media, there's all the scale up issues and things like that. So optimization is very difficult. Traditionally, people in industry, and I know this from being in the pharmaceutical and bulk chemical industries myself years ago, many people will just do random experiments or look at one of those factors, one of those 30 or 40 factors at a time. It's very inefficient to do that, and usually you don't get a truly optimal process. There are other companies that have started using things like factorial, fractional factorial design, or response surface methodology. These are methods that are out there, and you can buy software to do this. These are significantly more efficient than the methods on the left. But even these design of experiment methods, or DOE methods, are not nearly efficient enough when you have huge numbers of things that you need to optimize, and they tend to not use the developing information base very efficiently. So because of that, we've started using artificial intelligence to speed up the experimental optimization. And we're doing that using this approach that I've shown schematically here. The idea is that we don't want to just use an experiment's data to plan our next experiment. We want to accumulate all that information. In this case, we're doing that in an artificial intelligence model. And over time, that model of the process is going to get better and better and better. And so we can use it increasingly to pick where our next experiments are going to be to get to the optimum more rapidly. And that's what this diagram shows. If this kind of gives you two parameters, one parameter in this axis and one parameter in this axis, these circles are initial set of experiments. We would run the experiments. What you're going to see here is that over time, we're going to run more experiments, and that information is going to get better and better and better. And we're going to find out exactly where that optimum is and pick our experiments right around the optimum, which we're going to find in this case, in this example, simulated example, is right here. And the question is then, does this kind of thing work on cell culture media? And we did some experiments, just simple experiments with C2C12 cells, which are a mouse model for cultivated meat. We looked at DMEM media with FBS, fetal bovine serum. And what we found was, when we did those experiments, we did it both with traditional design of experiments and with our artificial intelligence methods. And what we can see on the right here is the growth media in red is the one using our method, and the blue one is the one using traditional methods. The yellow one is the media that we started with. So you can see we get faster and better growth with our methods. But probably the most important thing is we did this in 60 to 70 experiments with our method versus 100 using traditional methods. And those were even traditional efficient methods. So for those of you companies out there, think about if you could get to market in 30% less time or with 30% less experiments that are expensive, how important is that going to be to you? And I think you would say that's going to be hugely important. And that's why these methods, developing these kind of methods, is going to be very, very important. So what's next? Well, we're going from C2C12 cells to chicken cells. We've now isolated chicken cells, chicken muscle cells that we have as a stable cell line, which is great. We're working a lot with, and so we'll be doing using some of these methods with chicken cells now, and understanding how those different kinds of cells grow and grow differently than mouse cells and other types of chicken cells. We're starting to work with microcarriers, and we are focusing with other groups at Davis on edible microcarriers, because we think that's the direction to go, or it's completely to suspension culture. We're working on both of those things. We're working now we've adapted our cell lines to serum-free media, or at least C2, C12 cells. And so we're starting to do a lot of the optimization in that regard. We're trying to use inexpensive ingredients that would be agricultural waste streams to make the media less expensive, and we're starting to scale up. So these are all things that are going on at UC Davis with all our collaborators, and we're starting to collaborate a lot outside of UC Davis as well, throughout the United States and the world. So it's a very exciting time. Of course, with all this work, I want to acknowledge the people who actually did the work that I showed, Zach and Ted, and collaborator Keith Barr. I really need to acknowledge the great work that New Harvest and Good Food Institute are doing in funding research like this and promoting research, and obviously NSF for funding our research. I have my contact information there. I hope that now that these larger groups are starting to be set up, I hope there will continue to be support from these organizations and from you in industry. If you're interested in helping support these efforts and making them grow, please, again, contact me or these other organizations to have that happen.